And our next speaker is online, Dina Radenkovic. Are you there? We can hear you. We just need to be able to see you also. Wonderful. Just 1 second. Now we can see we can see your face now. Yes. Can you share it?
Let me try to share some slides. Okay.
There we go. Fantastic. Take it away.
Perfect. Okay, so I think we'll continue the conversation on the female reproductive longevity. My dear friend and colleague, Dr. Jennifer Garrison, has surely set up the final scene. I unfortunately had to miss the last pieces of her talk by waiting on in the queue. And I just wanted to thank again Alex, Morten, and all of the organizers for having us this year as well. My apologies for not being able to make it in person, but it's really, really fascinating to see how the field is growing and how many the brightest minds and scientists from all over the world are getting into longevity medicine. So the problem that we're all here to solve, and that our company Gameto, where I'll present some of our work during this slide, is trying to solve, is that ovaries are a faster aging organ. They age faster than the rest of the body, and we've already discussed how that relative decline can result in infertility, then menopause, and I'm sure Jennifer told you that menopause is a seminal aging event in a woman's life, and has an effect on her life expectancy as well as development of diseases of frailty. So we start back, my talk will be more around the therapeutics angle, because I'm sure that you're all aware that menopause is a burning problem that should be solved. So what we try and do, we go back to first principles. If the root cause of both infertility and menopause is essentially lack of ovaries, because we make them while still in embryo and we don't have them when we want to use them necessarily, let's try to make ovaries with science. So that's exactly what our company is based about. And it is, we go back to principles of ovarian cell engineering. So we have mechanisms that, using transcription factors and cytokines, we, from human iPSCs, develop and generate different type of oocyte precursors and ovarian supporting cells. And then we group that into the stem cell derived female reproductive system organoid, and then develop into 3 different lines. Fertilo, which is our assisted reproduction therapeutic that I'll touch on lightly, but then spend a bit more time on Diogo and Amino, where we hope to leverage the organoid of the female reproductive system, and then also the cell-based biologic for ovarian failure and potentially in the future, perhaps even menopause therapeutic, considering this is an aging conference and we should be talking about the moonshot to cure ovarian age. So this talks a little bit of some of our publications. Also, recently had 2 preprints that are under peer review in terms of the cell engineering mechanisms. So I'm going to lead anybody who's interested to perhaps review that, and I'll focus on to discuss very briefly, given that I've got about 15 minutes, the 3 programs. And then, so what did we do in assisted reproduction? The IVF process, we're going to start very briefly. I was so fascinated at the longevity medicine workshop to see neonatologists, physicians getting into medicine. I think studying embryonic development is so fascinating to address aging. But I'll just mention briefly, like, what's the near-term product of what we're working on here. Essentially, the assisted reproduction, infertility, 1 in 8 couples experience it. I think, like, people have discussed that it is a problem that needs to be solved because we are delaying childbearing for later years in this society. So what happens is that the cell extraction in order to perform assisted reproduction for men and women is slightly different, because the male cell extraction tends to be a bit of an easier process. Whereas for female cell extraction, to obtain sufficient eggs, we have to stimulate and give hormonal injections to retrieve the eggs, and many of them end up still being immature. So on average, women need 3 of these cycles, and then the success rate is around 40% of currently available assisted reproduction techniques. So what we do is that our solution was, okay, like, this process is not kind of great, and women are fairly medicalized to the process, and if we are doing cell engineering, and if we are engineering these ovarian supporting cells, could we use the ovarian supporting cells to derive a biologic therapeutic and then use it as a drug to mature eggs in a dish. So this is something that we are testing right now with fertility clinics in the United States and in Spain as part of the final stages of preclinical development. But we've developed a protocol in which women receive a reduced dose of injection, so around 2 or 3 days of hormones. Then their immature, smaller eggs are extracted, treated with our biologic, and then the eggs are mature and they can be either frozen or fertilized. The idea is that this could become a much cheaper, shorter process, allow more women to conduct procedures like egg freezing, which are currently the most effective way out of the existing interventions available to combat the age-related ovarian decline, but also mimics a more natural process and hopefully be more effective in the older age groups where women are certainly still very young, but the ovaries are already showing signs of biological aging. So yeah, I don't know if this video is going to work, but this is how it works. It's like a time lapse with like a real human egg, how it matures during the 24-hour co-culture. And considering this is an aging conference, we're not going to go so much into the creation of life, but this is something that we're, okay. But this is just something like a near-term program in terms of the therapeutics. And I think when we were looking in the company, assisted reproduction and finding infertility is probably a lower-hanging fruit in combating ovarian decline. It is already a success for science, particularly interventions around egg freezing and making that more commonplace. But certainly what we want to do as we grow and what we think should be necessary in the field, with more people really coming in and hopefully working in this space and working on the grants that Jennifer provides, and there are amazing other great companies that have been formed, like Viva Therapeutics of Cambrian and hopefully others, because there's so many problems here. And we need to really form an entire field to develop a class of therapeutics solving the phenotypes of female aging. We hope that perhaps the OVA platform could be a useful collaborator to enable some of that work. We essentially leverage additional cell types. So if you remember perhaps one of the first slides I've shown that we've made different types of ovarian supporting cells, but also oogonia, primordial germ cells, and cells expressing meiotic markers. So what we do here in the ovaroid, we say that essentially what is important for any drug development is a good model. And I think we've seen with models like pulmonary fibrosis that it was almost a graveyard for drug development because we had to burn the 2 layers of the pleura with bleomycin. Well, similarly with ovarian aging, Jennifer certainly told you that it's humans and 4 types of whales that experience it in the same way. And the way we induce it in even primates or murine models is by damaging their ovaries with a chemotoxic agent. So we hope that it is something that was done with a sponsored research agreement with George Church's lab at Harvard and the Wyss Institute. But we hope that with our first stem cell derived organoid, we're able to be an early tool in the very early stages of perhaps disease modeling and drug development. Now, one of the questions that comes up in using anything derived from stem cells for disease modeling or testing is the epigenetic age of the stem cell derived tissues, which is why we have established an organ donation program with Johns Hopkins, where we obtain for research purposes cadaver organ donor ovaries, where we are able to. So this was in our lab actually about 2 weeks ago. So we were able to obtain from different age cohorts human ovaries and do bulk RNA sequencing in order to adjust potentially computationally and then decipher the molecular signature of an older ovary of a 64-year-old woman versus a younger ovary, and then see how they're different, and hopefully for the future provide a platform that we could perhaps even have a small molecule that changes the methylation profile and rejuvenates or removes the damage accumulated in the ovary. This is not something we're working on therapeutically right now, but creating a knowledge platform so that us and partners working in this space will be able to create. So we hope that in the future, our stem cell derived organoid can not only do things that we can do right now, which is just do a very basic tissue toxicity before you, let's say, recruit a woman in a clinical trial and check how this affects her ovarian reserve, but hopefully model different diseases, because again, female reproductive system is very, very underserved area. And we have conditions that are as common as diabetes and affect big portions of populations that don't even have first-line medications. And obviously, I think with all of the hype that initially came in the organoid work, recently we have seen that they can be early tools in the early stages of drug development, and many big pharma companies signing deals in research, business development, perhaps partnerships with some of the mid-stage biotechs working in this space. So hopefully will allow us to form more partners and start building the knowledge base for some of the unknown questions of the diseases of the female reproductive system. And then I'll finish off with our third program, and this is something where it's really a moonshot project, which starts questioning, like, is this ovarian aging that we're experiencing even like meant to happen? Like, what if it didn't happen? Could we dissociate the loss of fertility with this whole development of frailty and experiences that occur to women during perimenopause and menopause? So Jennifer has already told you the whole story, but it is a huge market of 1 billion women, and I think like there's a lot that we can do to improve the health and start building novel therapeutics. Current hormone replacement therapy is probably the best that currently exists, but it certainly has side effects and is far from ideal, and it does not cure a lot of the problems, whereas some of the currently existing selective estrogen receptor modulators can treat conditions like osteoporosis, but again, not immediate side effects. So how do we came up with this idea? We found it from a real clinical example. So this is a story that we learned in clinical medicine for fertility preservation of prepubertal girls. Often, this is something that's been done in the UK for longer, but even in the United States is no longer considered experimental procedure, where essentially prepubertal girls are obviously not ovulating, so they cannot go for egg freezing, which would be a standard of fertility preservation before chemotherapy for older women. But what they would perform in these girls is they would perform a more invasive procedure of ovarian biopsy, and they would then slice their ovarian cortex into different patches, freeze, and then later on implant the tissue patch either near the fallopian tubes, so back in the abdomen, or occasionally at the anterior abdominal wall, or even in the part of the forearm. So highly vascular areas where you would perhaps wear your continuous glucose monitor. Now, what they've learned from these patients with obviously their own autologous cells is that many would conceive naturally, even without the need for assisted reproduction, because their own cells would essentially integrate into the negative feedback loop between the ovary and the brain, responding to the FSH signals, tricking the brain to think that the physiology has restarted. What some clinics have done is that they have said, okay, like if essentially this works, could we start offering this to women as a menopause prevention procedure? So this is a controversial area. It's not something that we work in, but at our clinics, if you go online and say, like, could I perform a procedure to cure my own menopause, they would perhaps offer you to have an ovarian biopsy during a C-section, and then have your own ovarian cortex slices frozen and then perhaps reimplanted to delay menopause. So it is in clinical practice for fertility preservation, and then it is in a controversial area for menopause prevention. We don't yet know if it works. But we then said, okay, like if we have like a cell line that is allogenic and it produces the hormones that control female physiology, and if it responds to the FSH, could we essentially replicate something like this, but provide it at scale and build it into a therapeutic that could really, again, dissociate the loss of infertility with all of the side effects that occur during menopause. So trick the brain that it's not going through this process. And this is something that we have seen, right? Like cell-based therapy is becoming mainstream. We're seeing that engineered pancreatic-like cells are curing patients in clinical trials for diabetes by Vertex, also therapy license from Harvard. We're seeing with CRISPR curing sickle cell anemia and beta thalassemia. We're seeing already in the clinic, successful example of CAR T-cell therapy, and many biotechs curing different types of blindness with cell-based therapy. So it is no longer just science fiction. It is perhaps becoming real science. But you would argue that unlike perhaps conditions like diabetes or cancer or blindness, ovarian failure is perhaps not considered as severe. And with breast cancer being the cancer that affects 1 in 8 women, why would you ever like to have a cell line that is engineered that could produce estrogen? What if you develop an estrogen-dependent cancer? And you would be correct. Probably people would say, look, the cancer risk, the C word is so big, I would rather go through menopause. Which is, again, a big reason why many women do not undergo and do never take hormone replacement therapy, because of the fear of increased cancer risk that has been reported in the Women's Health Initiative study. So what we then said is that, okay, so if this product was to ever become reality in clinical practice, it would have to be something that has an enclosed space. So our current prototype is actually a rod. We initially thought it could be a patch, but guess what? Thanks to it's science, not every idea works on the first pass. And what happens is that the cells will be in an enclosed environment, and the way the product will be inserted, it will be inserted in the subcutaneous fat path in the arm with the small cannula. And it would have to be because the cells are obviously in an enclosed space, replaced every 4 to 6 weeks. So it becomes something that women can put in, perhaps could be a cure for primary ovarian insufficiency / primary ovarian failure, early menopause that occurs in young women, but then later on could perhaps be licensed for control of menopause-related symptoms, the way the currently hormone replacement therapy is licensed. So we're kind of getting to okay, how could this product work? And the beauty here is that because the cells are FSH responsive, they would produce a personalized hormone delivery for different women that apply it, and trick your brain to think that, okay, I am not going through this process. There's no this crazy FSH upregulation in the brain that has recently been reported to even associate in mice with severity of things like Alzheimer's disease, right? There are a lot of potential consequences that occur after this seminal aging event. So it dissociates loss of fertility that occurs in menopause and loss of periods, with potentially some of the downstream consequences like osteoporosis, but also the abrupt hot flashes and other things women experience. In any case, that's kind of a little bit of our run through of what are the therapeutics in the area that we're working on. We are a company using cell engineering to develop novel therapeutics for the female reproductive system, with headquarters in New York, offices in Madrid, and in collaboration with George Church's lab at Harvard Medical School. And we really hope that the cell engineering platform would not only enable some of our near-term work in assisted reproduction, so combat infertility, the first phenotype of ovarian aging, but that ourselves or together with partners or more people coming into the field will be able to tackle the later stage phenotype of ovarian aging that is currently vastly underserved, and it involves menopause and the diseases of aging that occur afterwards. So I would like to obviously thank our brilliant team of scientists who are working really hard to make this happen, our scientific advisory board, and everybody else who's contributed to some of the work on these slides. And I think I just finished in time so that the next speaker starts in time, and if maybe we even have time for a question. It was very helpful to be after Jennifer. She is the thought leader and an expert in this space, so I could skip through most of the background. Let me just see, how do I...
All right, thank you so much, Dina. Can you hear us?
Yes, I can hear you.
Okay, fantastic talk. I think we have time for 1 or 2 questions. We have down here in the...
Thank you very much. I think it was extremely interesting and important subject. I do think that it is, first of all, you need to separate between fertility and menopause, which are 2 different things. Although it is important, and we know that the menopause incidence when it starts has a correlation with aging, but still there are 2 different areas. But I do think that in a longevity, Menopause is extremely important. At least 50% of the population are women, and they will age the same as men, even though we die a little bit later. But it is important to emphasize, and especially taking supplements or estrogen, and to try maybe not to take it artificially, but to work on what you are doing, trying that the body itself will reproduce it, which is will be better than taking it from outsource. But I think, and especially in the longevity center that we are going to open, which will be clinically with the clinical application, main part will be menopause, which is will be integrate into the algorithm, and I think it is extremely important, and it should be done in every longevity area. Thank you.
Thank you very much.
Yeah, absolutely. I thought, like, I think I know. I think I know who the question came from, and I was extremely impressed with your presentation at the longevity medicine workshop, and really fascinated that you were able to share the story. And I think certainly menopause is kind of a taboo topic, you know, like women, we haven't really spoken about it so much, and I think using even the existing interventions and see how they can be repurposed to use them better will be important, and it should be part of every preventative and longevity medicine center.
All right, we have one more question from Alex.
Thank you, Dina, for such a great presentation. I'll ask, but before I ask it, I wanted you to maybe sit with a different angle, because you're sitting, the window is behind you, so it's impossible to snap a good picture. We cannot see you. So yeah, if you can just change the background somehow, that would be great.
I'm not sure. I apologize. I'm actually taking this on an iPad, so I'm gonna,
because I want to promote it, right? But anyway, so while you're doing this, I wanted to ask, what are the major challenges? You're a very successful young female entrepreneur who started, well, who is a CEO of a very successful young startup. What were the main challenges for you in, finding this company and in this company? What are the current major challenges for female reproductive longevity and equality in the startup world?
I mean, it's a very complex question that goes far to company building. I would say, like, it really is that we haven't had too many success stories of profitable companies in this space. And the way we structure Gameto, I was a partner in the biotech fund, and I had the research post, right, like after I exited the longevity clinic, was really something that I think is relatively investable. And we kind of narrowed down, going from the first principle, is that a single cell engineering platform, and essentially the same cell lines, just slightly tweaked, could be applied to solve several conditions, right? But then you cannot solve too many problems at once. So how do you sequence it? Nobody is going to really give you, I'm going to, give me a lot of money and then I will cure menopause. It's a moonshot project, right? Especially in the current market, I think we have this push to be, what is your time to product market fit? What is your time to profitability? So the way we structured it is that our first product, where we spend 70% of the resources as a company, is really in leveraging our technology for assisted reproduction. Why? Because it's somewhere where we very early were able to get preliminary efficacy. We're in preclinical development. And secondly, it's the regulatory in getting something that is delivered outside of the body, and it is much faster, right? So we have a product that we can, as a small company, essentially show traction and bring to the clinic, while enabling and then showing people that we can deliver, allowing you to then stage and prepare you for your next goal. So I think creating near-term and long-term goals is particularly important in order to prove that you can make you can raise sufficient capital for the things that you want to develop. And I think if Gameto is to be successful, and we're obviously trying really hard to make that happen, we want to ensure that our products are serving women, empowering women, are products that people love and want to use, and that are fairly profitable. Women account for 83% for healthcare spending, and I think we had just a lot of innovation in digital health and service delivery, but not in biotech. So I really hope that that is something that we can prove, and then I think there will be a lot more funding in this space that will just come naturally.
Perfect. Thank you so much, Dina. Fantastic talk. Let's give her another round of applause. Really great. So.